So to start, I really wanted to emphasize the importance of our microbes, not only in the case of disease, but also in establishing our health. So what I'm going to tell you over the next 30 minutes or so is really about how among our microbes we can find friends, rarely foes, and for the most part, just find microbes. So these are the majority of microbes. They're not really either good or bad, but they're there for a reason. And we're going to talk about how all of these affect inflammatory bowel disease. So what I hope is that by the end of this presentation, you'll be able to really appreciate the world of microbes. But to start, I have no disclosures other than really to preface my presentation by reminding you that we're all covered with microbes. And of course, right now we're doing a pandemic, and you probably don't need to be reminded of this. But it's really a good time to also remember that the vast majority of these microbes do not care at all about us. I personally find it really incredible about how our lives have really been upended by a microbe so small, COVID-19. This microbe is about 7.5 million times smaller than us. But I'm here to tell you about how we actually need certain microbes, and in fact, how they can protect us from infection. It really doesn't seem like it in 2020, but infectious diseases are strongly on the decline. Over the past 75 years, the development of vaccines, antibiotics, and new drugs, as well as improved hygienes, has allowed this unprecedented reduction in infectious disease. And this really has correlated with the incredible growth of human populations that we've seen. One of the challenges is that mirroring the sharp decline in the incidence of infectious diseases, there's also been this alarming surge in autoimmune and allergic disorders. In the 1990s, scientists began to suspect that these two trends were connected, with the idea that perhaps the reduction in infections was causing the human immune system to malfunction in some way. And we're now finding out that this is indeed the case. The problem really lies in the fact that when we combat microbes, we usually are not selective. And what the problem here is that we're not just fighting off the pathogens. The problem becomes that we're starting to kill off a lot of the microbes that live within us. And some of these microbes are really important for our health. So the reason behind why we don't want to get rid of the good microbes or the neutral microbes is really going to be the topic of the next few minutes. And one of the things that it's been really discovered over the past 50 years is how our microbiota is really a microbial ecosystem. We have inside us and on us tens of trillions of bacteria, viruses, and fungi. And these make up what we call our microbiota. So our microbiota is really the microbes that are associated with us as humans. And they are really important for our health. And not only are there so many, they also come in a lot of different bacterial species. Each one of us has hundreds of different of these species. And you may wonder why we actually need the microbiota. And there's a lot of different reasons why. First of all, we need them because they help digest our food. Depending on your diet, they can make up a really large fraction of our caloric intake. We have also evolved with these systems, and they really help us regulate our immune system. Importantly, they also protect us against pathogens. And this is not really out of any type of generosity, at least that we know of. Rather by the fact that because of ecological principles, they actually occupy some of the niches that once they're filled, they will keep the invaders out. They also produce vitamins that are really important for our health. Some examples here are vitamin B, K, or riboflavin. One of the things that is really exciting for us in terms of studying these systems is how diverse they are and how many genes they have. So the microbiota as a collection has this incredible functionality. They have over 150 times the number of genes that we do. And so they can potentially have many more functions. And I'll tell you now why this matters. When you think about what happens when you take a pill by mouth, is that the compounds get released into our gut and they make it through our entire body. The microbiota produces compounds that go into our bloodstream the same way, just by crossing the intestinal barrier and making into our blood system. So the microbially produced products can really affect anything from our gut to our brain to neurodegeneration to really all of the normal processes that happen inside us humans. And importantly, microbes also modify and respond to specific drugs. There's a lot of evidence now For example, how different people, depending on their microbiota, can have different response to cancer response therapy. They will modify the tumor microenvironment, and they will affect the way that certain drugs are able to work or not. There's also, of course, effects in IBD as well. Even the response to biologics is stratified based on the underlying microbiota. And importantly, who is part of our microbiota dictates what drugs are changed and what bacterial products will end up getting into our blood. In turn, what dictates who is there is dependent on what we feed ourselves and therefore how we feed our microbes. So the question then becomes, what are we feeding our microbiota? And this has changed incredibly over the past hundreds of years, as we have modified our diet from a more traditional one to one that is more industrialized. So traditional populations, even nowadays, for example, the Hadza that are pictured here, have a diet that is very non-processed and is very rich in fiber. Conversely, what has happened to us is that as industrialized populations, we have developed types of foods that have much more easily accessible nutrients, that are very highly processed and that lack fiber. And they really lack the type of food that our microbiota needs to survive in its initial form. Here, what is very important is that there really isn't anything that is fundamentally wrong with an industrialized lifestyle. It's just how we respond to it that is problematic. And this really goes back to evolutionary timescales. Here, this is a picture that I really like a lot by Banksy that shows a primitive person that is eating McDonald's. From the perspective of our own evolution, we have evolved with our microbiota ever since animals have had a gut. That was over 548 million years ago. And during this time, we have also evolved with a non-industrialized lifestyle. The difference here between how our microbiota responds to how we respond to changed lifestyle comes from the fact that our own human generation time of having their next offsprings is in the order of 20 to 30 years. But for bacteria, it's 10 million times shorter. What this means is that upon changing our lifestyle, it doesn't take very long for the microbial species that are not doing well in that new environment to become eradicated. But in the meantime, our own human biology hasn't had the chance to adapt. What this means is that in the end, our biology may be mismatched to our industrialized lifestyle. And we know that this also happens very strongly through our microbiota. What happens when we mismatch our lifestyle and our microbiota to our biology is that we start seeing diseases that were completely unknown to our ancestors and that are still unknown to traditional populations. So here you see some of the ways in which we've changed our lifestyle. They come from consuming specific types of drugs or antibiotics, having changed diet, even how we're born or how we raise our children affects a lot how their microbiota is able to establish. And these changes have been correlated with a lot of different diseases, ranging from allergies to immune disorders, and of course, also inflammatory bowel disease. One of the challenges with IBD is how heterogeneous it is. So by heterogeneous, I mean that it's different from person to person, from patient to patient. And this is really the underlying reason why we don't have a cure yet for IBD. We know that there's a lot of factors that are involved in IBD, and some of them are very hard to change. For example, our genetics and our immune responses are so dependent on our history that it's hard to change them at the time of the diagnosis. But fortunately, there are things that we can change and that we can influence. And some of these are our environment, so what foods we eat, whether we smoke, whether we use different types of drugs, as well as our gut microbiota. One of the things that I'm really excited about in terms of the gut microbiota is that through it, we really have a chance to do personalized medicine. And this is the reason why I'm in the field, is that I really believe that the microbiota is going to be a really important way in which we can tackle inflammatory diseases. So my goal here then for the rest of the talk is to really tell you about how there's different studies that we're doing that are looking at how personalized medicine can really affect the microbiota and then affect inflammation. Here first, let me tell you a little bit about what our goal is. So the goal is with microbiota therapies is to change what is called the inflammatory microbiota to one that is not only non-inflammatory, but is also resilient to negative influences, and that might make it become inflammatory again. And here are some of the key discoveries and characteristics that we have discovered in the field. I mentioned that there's hundreds of species in the microbiota. What happens in an inflamed gut is that this number of species becomes much smaller. So the lower diversity means that not only these microbes make fewer compounds, but also they're less resilient against invasion. Here you can really think about it from a perspective of an ecosystem at the macro scale. If you have a forest in which you've decimated a lot of the native trees, it's going to be much more susceptible to any type of infection or any type of invasion by other species. Here, the other two words that you see is facultative anaerobes, and these are increased. These are bacteria that are able to tolerate oxygen. Oxygen is something that is not normally present in the gut. The gut is actually anaerobic, meaning that it doesn't have any oxygen, and this is a normal and non-inflamed state. What happens during inflammation is that the gut produces a lot of oxygen, and this makes it through the gut from the intestinal tissue. So these facultative anaerobes are bacteria and microbes that in general are able to tolerate the oxygen. And these are microbes that also tend to sometimes promote this inflammation because they're trying to foster their own growth. The goal here now with microbiome therapy is to lead the system to have fewer of these facultative anaerobes and to really encourage the growth of the anaerobes, meaning that these are the oxygen tolerant species. These are the species that tend to be very good at fermenting fiber and that produce compounds that actually reduce inflammation. Here now I'm going to tell you about how we can follow our goal to reduce inflammation and how we can have different types of microbiota therapies and how they can help. I'm going to highlight a number of different microbiota treatments that are showing promise. And this data is now that I'm showing is from a great review from the Xavier group. And I will talk about different therapies and explain whether they have a direct or indirect effect on the microbiome, on the gut metabolites. So these are the products that bacteria make and release into our gut, and as well as whether it reduces inflammation directly or indirectly. And here you can see the keys that the direct effects are in dark green versus the indirect ones are in light green. The first is really what is the current standard treatment for affecting the microbiota is changing enteral nutrition. And enteral nutrition generally refers to any method of feeding that causes the gastrointestinal tract to deliver the caloric requirements that we have. And in terms of the gut microbiota, really the emphasis is on non-processed foods. And foods that we often talk about the Mediterranean diet, so foods that will really have a high content of fiber as well as a well-rounded type of diet. One of the things that is also talked a lot about and is currently in a lot of critical trials is this idea of transplanting microbes. So fecal microbiota transplantation affects directly the gut microbiome and as well as the gut microbiome metabolites. And it's really the transplant of the microbes from a healthy person to a patient. There's a really great talk about this in one of the Gutsy Learning Series talk regarding fecal microbiota transplant. So I'm not going to talk a lot about it. It's showing promise, but it's also showing challenges. In some of the ways that we've seen it work very well, for example, against Clostridium difficile, it can be really targeted and work very strongly. But again, because IBD is very heterogeneous, it seems to have require specific ways of working. So I'm also going to talk about how, with our own research from our lab, how we can think that we can make it better and make it more targeted. Next, I'm going to talk about 3 other types of ways in which we can influence the microbiota. And these are things that you probably have heard a lot about. These are the prebiotics, the probiotics, and the postbiotics. So these are things that have been tested either in animal models or even in clinical trials. And there's a lot of buzz about pre, pro, and postbiotics. And a lot of what you'll find on the shelves of the supermarket is not very likely to have been thoroughly tested. So I wanted to spend a little bit of time telling you about what you really need to know about them. So prebiotics are specialized fibers. They often also come as supplements. A key point here is that whether you're feeding them to the microbiota is going to work only if you have the right species to feed them to. And this can be problematic if as industrialized populations, as we're starting to lose a lot of the species that, as I was mentioning before, that we have evolved with. So it becomes really important that prebiotics are also in being targeted with specific microbes that are going to be there and going to be able to take advantage of this newly found fiber. And we're really working on trying to understand this. Probiotics, on the other side, are the actual bacteria. And one of the questions about these probiotics is, who should we really be adding? One of the challenges right now with probiotics is that they're often coming from species that have been isolated even sometimes decades ago, and they were isolated because they would grow easily. And there's still a lot of testing that needs to be done for species that will actually be positive, not only for specific indications, but really broadly. And one of the things that is important is that also probiotics come in different types and different flavors. So the type of probiotics that you'll find in yogurts or kimchi, those are much more natural types of probiotics as opposed to what you'll find in a probiotic pill. So for the most part, probiotic pills have had mixed results. And there's still, again, a lot of research that needs to be done to really target what we call the mechanisms of trying to understand how they work and whether they can be beneficial for different patients. Postbiotics are what the microbes actually make. And this is an area that is being really actively developed, trying to understand whether there are specific molecules that are made by the bacteria that we can add into people, so that even if they don't have the specific bacteria that are missing, they can help alleviate the inflammation. So the important thing here is that, unfortunately, with the diseases that are very heterogeneous, such as IBD, it's really going to be hard to find a one-size-fits-all solution. And what we really need to, and where we're trying to go towards, is trying to understand how, for different patients, how these different probiotics or prebiotics can really make a role in reducing inflammation. Another type of microbial therapy is targeting the microbial foes. So there's some bacteria that tend to not respect the boundaries between ourselves and our gut digesta. And these are bacteria that try to make it through the intestinal lining and cause a lot of inflammation. So, for example, an example of this is adherent invasive Escherichia coli. And there are ways in which the attachment can be reduced. And so there's a lot of therapies that are being developed right now to try to prevent this inflammation caused by specific pathogens. Now I'm going to share with you a specific type of postbiotic that we have been working on and trying to see, this is in animal models, whether it can be a good source of reducing inflammation. So here I'm going to tell you about another way in which the microbiota affects our health. And this is by transforming nutrients that are being made by our body. So the microbiota is able to transform bile acids that are made by the liver, and it transforms it into a number of different bile acids that are called secondary bile acids. So this can be cycled back to the liver for additional modification or excreted as waste. This is a really good two-way interaction because bile acids provide an energy benefit to bacteria, but they can also reduce inflammation. So what we have found is that the secondary bile acids can be really beneficial in the case of colitis. So in a recent study that we did in collaboration with the Aptesian group at Stanford, we showed that ulcerative colitis patients that had undergone colectomy, so this is the removal of the entire colon, had less secondary bile acids compared to patients that had undergone the colectomy for another reason. What we then started to try to understand was whether the supplementation of secondary bile acids would be able to reduce the inflammation. And what we found is that in 3 different animal models, they were able to do so. And I'm going to show you some of the data. So during colitis, what we find is that the intestinal tissue is very inflamed. What you see normally is that the intestinal tissue is relaxed instead of having this globular formations that you see here. And what we did was to add secondary bile acids, and you can see here that the intestinal tissue looks elongated and allows a better absorption, and it's just generally not inflamed. And we found that we could make these changes with 2 different types of secondary bile acids. And importantly, what we found is that the modification done by the bacteria were really necessary, because when we added primary bile acids, and these are the ones that are just made by us as humans, they really do not reduce the inflammation. So what is really needed is having the secondary type. So what really this told us is that the influence of these microbes is really important, and that there are molecules that we can at least add in these animal models to really target inflammation. And we're now starting to try to understand, the Abdesian group is working further to try to see if this can be also adapted for human patients. So far, I've told you about how microbial therapies can ameliorate IBD. And now what I'm going to do is to tell you how IBD changes the microbiota, and how that in turn can be problematic for our health. So to start, I would like to tell you a bit about how we study the microbiota. One of the ways that is most common for studying the microbiota is by sequencing. And sequencing tells us what microbiota members are there. The other type of measurement that is very important is called metabolomics, and this measures what are the bacteria making. One of the ways in which we're trying to explore the question of how the microbiota functions in a bit of a different way is by adding imaging to this type of approaches. So if you've read any popular articles about the gut microbiota, you will have heard about how sequencing is one of the main ways of interrogating the microbiota. And really, this allows you to count the different species that are present, but it gives you a very reductionist picture about how a living system works. So here, I think if you have work to explain to you how a tree works by enumerating the types of branches and pine needles that were present. What we really want to do is to understand how they fit together. So in our lab, we complement the enumeration process with measurements of the microbiota structure, and by measuring also the molecules that are being produced by the microbes. Here now, I'm going to show you what it looks like. So this is an image of a gut section, and this is will be the same by looking at a biopsy. And you can see that there's a lot of structure that is being present here. The structure of the ecosystem is really important because it really defines their stability and their resilience. Let me give you just a little bit of a map as to what you're looking at here. At the top, and highlighted in blue, these are the intestinal cells that make up our gut lining. What our intestinal cells do is to produce this mucus, which is in green, and they produce it and push it out. And this allows us to keep our distance from our microbes. But importantly, it also keeps the microbes fed. So our mucus is keeping us away from our microbiota so that we're a safe distance. But in case we don't eat our fiber, let's say because you had a burger instead of eating your veggies, it can sustain our microbiota and feed it over short time scales and over the short term. One of the things that we wanted to understand is what happened during some of the common symptoms of inflammatory bowel disease, specifically during diarrhea. One of the types of diarrhea that we study a lot is malabsorption. So this is when there's compounds that are not being absorbed properly, and they cause increased flow. This, of course, is also what happens with celiac disease, and also what is the mode of action of a lot of laxatives. So normally we have the situation in which when we have, this is again in animal models, when we have these untreated animals, we see that the mucus lining is thick and separates well our cells in blue from our bacteria, which are in red. What we were really surprised to find is that during diarrhea, this protective mucus layer was completely and almost completely depleted. What we found that this led to is the direct contact between bacteria and our cells. And this is something that our cells do not like very much. This leads to not only an immune response, but it also leads our system to try to fight bacteria that normally are known to be good for us. And these findings were really important to us because they were things that we could have not have understood just by sequencing. And they really gave us insight into some of the ways in which by by changing our gut environment, we can also affect our bacteria and the way we can interact with our bacteria. We think of diarrhea as acting acutely, and we do indeed find that the environment seems to recover at the end of the treatment. And at that point is then when we asked, does the microbiota survive this diarrhea, or is it affected for the long term? What we find here is that diarrhea indeed creates a disrupted environment, and what we find is that this disrupted environment can be exploited by pathogens. So pathogens, of course, are trying their hardest to survive, and what they're better at than our neutral or good microbes is the fact that they're really good at surviving in stress conditions. That's why it's so hard to get rid of them. Here I'm showing you a plot that shows the abundance of specific bacteria. So 0.1 will be 10% of the abundance of all the bacteria. And this is looking at a time course. These animal models had 6 days of mild diarrhea. And during the time of mild diarrhea, you can see that there's an increase in this types of bacteria that are called the gamma protobacteria. These gamma protobacteria are your typical E. coli or Proteus, and a lot of these species that tend to be more pathogenic. And when we find them increasing, we also find that this commensurate reduction in other types of bacteria that tend to be the ones that are potentially more protective. We also found that diarrhea can cause long-term changes to the microbiome. And here I'm going to focus on the presence of two really abundant gut bacteria. And these are bacteria that come from a human microbiota. So one of the things that we can do in these animal models is that we use microbiota that come from human patients so that we can really study how the specific human bacteria are going to be affected by these different perturbations. So in this meeting, there's about 30 people that are listening to this that have this family of bacteria called S24/7, and everyone listening to this have Bacteroidecia. You can see that these are bacteria that in our control samples are quite stable, and they make up a very large fraction of this human microbiota. So S24/7 specifically starts out at almost 50% of the microbiota. And what we find is that during diarrhea, and we found this really surprising, is that this entire family of bacteria became undetectable. And you can see here how its disappearance is overcome by an increase of the Bacteroidecia. And here the Bacteroidecia are taking advantage of this newly opened up niche. What was even more surprising to us is that when we took away the agent that was causing the diarrhea, we found that S24/7 just never recovered. And here I'm going to stop. We would have never expected that malabsorption over 6 days could cause the disappearance of 50% of the microbiota. We're talking about tens of billions of bacteria. And not only is this a large number, we actually cause the extinction of an entire family of bacteria. To give you a sense of what it means from the perspective of an ecosystem, this is akin to, if we're talking at the macro scale, if we got rid of all felines or all primates from an ecosystem. This is a really huge change. And we're really interested in understanding whether S24/7 is important to our health. And we're starting to find indication that it's really involved in our immune system regulation. So these are two studies that have come up in the past couple of years that are showing how S24/7 is really tightly linked to our immune system and in the way that our immune system is able to respond. So we're currently really trying to understand the importance of S24/7 in inflammation. But one question that we were able to address was whether we will be able to restore it. We were really interested in understanding whether we could restore this disappearance of key microbiota species. And I'm now going to tell you about how we can go about getting them back. Here I'm going to show you other experiments. Again, we have the abundance of the specific bacteria, and we're only going to be looking at S24/7 specifically. So in these experiments, again, S24/7 would respond really negatively to diarrhea and became undetectable. And then we had a fecal microbiota transplant from a healthy and controlled microbiota. We really didn't know what to expect because, as I'd shown before, when a niche opens up, it is usually filled up. And we were really excited to find out that after this fecal microbiota transplantation, we were indeed able to reestablish S 24/7. And it was a really profound finding that a steady state community could be recolonized after weeks of not having had a specific member that was part of it. But what we learned that was even more important is that there's specific requirements for this recolonization. Importantly, reprogramming the microbiota requires a physical environment that is permissive. So what we asked in these experiments now is, when did it matter that we reintroduce S 24/7? So on the experiment on the left, we tried to reintroduce it during the time of diarrhea, while in the second experiment, we kept trying to reintroduce it for a couple of days after the diarrhea was done. And as you can see here, only when we had this addition continuing beyond the time of diarrhea were we able to reintroduce this. And importantly, this really showed us that, as you might expect, again, in thinking about a macro system, that the timing of this therapy really matters. We need to have a physical system that can accept these microbes in order to be able to transplant them in. And we think that this is going to be really important for microbiota therapies, because the fecal microbiota transplants are not going to work as well, we think, during times of flare-ups, because the physical environment is not going to be able to really accept these different microbes. In terms of thinking about how this fits with the existing IBD therapies, we don't currently have an option to improve the gut environment. And we really believe that this is a really critical step for microbiota therapies to work. So our vision is that before microbiota therapies, we need to restore the physical environment of the gut. And we believe that even in the case of thinking about prebiotics or probiotics, that we need to be able to accept them into our ecosystem. And importantly, this is going to have to be a personalized approach. We know that in IBD, there's changes in pH, there's changes in the case of diarrhea, and we need to understand how different people respond and how their physical environment is changed. Once we can restore the physical environment, we can then allow the recolonization by beneficial microbes. And really importantly, once these beneficial microbes have come in, we really need to sustain them. And this is where we think that diet really plays an important role. To conclude here are some of the messages that I hope that have come through from this presentation. So first of all, we're home to 10 trillions of microbes, and these make up our microbiota, and they're really essential to our health. Through industrialization, we have changed our lifestyle as well as our microbiota, and our microbiota is able to evolve much faster than we are able to keep up with. And we think that this has come up with this very heavy burden of inflammatory diseases. And our goal is really to use microbiota therapy to restore a healthy gut. Finally, I've told you that we have many different types of microbes. We have our friends, which are the healthy microbiota that we really need to feed with good fiber. And one of the things that over the years that I found to be most useful is that if you want to know how much fiber is in a food, you can just Google this and Google the name of the food and write how much fiber in the name of the food, and you'll find out which type of foods have fiber. And there's some foods that you might not imagine that have so much fiber. For example, avocados have a ton of fiber. And generally, to sustain a good and diverse microbiota, we really try to have as diverse a type of fiber as possible. And of course, different people respond differently to different types of fiber. So here it's really important to consult with a dietitian as well as with your doctor to make sure that whatever diet changes you do will be important for you. We also talked about foes and how gut disturbances can facilitate pathogen growth, as well as how there's different ways in which we can try to target these pathogens from not attaching to our intestinal lining. And then mostly we talked about these just fine microbes. These are the ones that we have evolved with, and so many of them are important for our health, and we really need to keep them alive and producing these compounds that are important for us. To end, I would like to thank my group for pursuing a lot of this work. I would also like to thank our collaborators at Stanford, Dr. Abdazian and Dr. Sina. And we're really grateful to our funding agencies, and we're really grateful for Crohn's and Colitis Canada. Without them and without your donations, this academic research would really not have been possible. And what is really important is that right now we need the research. There's many more questions than we have answers. And one of the things that also that has been really great in connecting with some of you through Crohn's and Colitis, and I know that a lot of you and a lot of your family members have really suffered a lot because of IBD. And I guess one of the things that I hope that you can take from this talk is that there's a lot of us that are trying really hard and really working hard at trying to understand how can we make this better and how we can find a cure. So we really appreciate your support and your involvement. And thanks so much for listening to this talk, and thank you for supporting us.
Thank you so much, Carolina. That was fantastic. I learned so much. The next phase of this presentation will be the question and answer period. What we've done is the audience members who were registered at the time of registration have the opportunity to submit questions. So what Sarah has done is compile those questions and group them into themes. So we have slides with those questions. I see the audience members as well are typing in questions in the chat box. So hopefully we'll be able to go through the questions that are in the chat box as well. But maybe we can pull up the first question, Sarah. So the first question submitted by the audience members at registration time was, are there any treatments for IBD being studied that target the microbiome and are effective? I know you spoke a little bit about that, but perhaps you can elaborate.
Yeah, absolutely. And you know, the question about natural treatment options, I think it's a really important one. Probably the most natural treatment that we can think about is changing our diet, in changing the way that we support our microbiota. So this kind of goes back a little bit to thinking about whether the Mediterranean diet it works for everyone. That is not necessarily the case. But I think that trying with different diet options is one of the ways, one of the initial ways that I will go about natural treatments. In terms of fecal microbiota transplants, I touched upon it a little bit, and here it looks like you also have a link to the Guts Learning Series talk that I would highly recommend for people to think about too.
Great, thank you. Next slide, Sarah. Great. The next question submitted was, are probiotics effective in maintaining or promoting a healthy gut microbiome? I guess, again, something that you touched upon, but if you can elaborate, and I see there's some sub-questions linked to that as well.
Yeah, absolutely. So probiotics are a bit of a controversial, I guess, a treatment right now. I think they will hold a lot of promise in the future, but right now we need more research about them. And here again, I would like to go back to this idea of this natural ecosystem. Like if you think about, there's a forest that is having problems, is undergoing desertification, and the question of if I add a pine tree, will this help? If you think about it at the macro scale, it becomes really obvious that it's going to be hard to have a single species that will help restore an ecosystem. And one of the challenges too is that a lot of the probiotics that we look at are often in doses that are really, really massive. So there'll be billions of the same microbiota member that you're going to be taking down. So that I know of, there's not really evidence that probiotics are negative for health, but they've been really spotty in the ways that can be beneficial for health. And then one of the ways in which that I would also like to comment on is that for probiotics, there's some new research that, for example, in recovery from antibiotics, they actually seem to hinder the normal return to the previous microbiota. While, for example, oh, sorry, Katie, were you going to say something?
No, I just said, I didn't know that.
And so that research got publicized a lot, saying the probiotics were bad, and I think that's too much. I think that there is a place for probiotics, but I think it's going to be unlikely to be one species is going to fix everything for everyone. What I think that we need to restore, much like a natural ecosystem, is all of the species to kind of come back together. We need the diversity, and I really like to think about this as a forest, that we need to make sure that it's not just going to be a monocrop. We need to have a lot of different species that come together. In terms of particular brands, of what I can speak to, I'm not a medical doctor, so I can't give medical advice, but some of the research supports BSL-3 probiotics as having positive effects, as well as also the Coli Nissel, that have had multiple studies that have had some positive effects. But also here, it's not with all of these treatments, it's going to be really dependent on the specific person, because we know that different people react differently to different types of fibers, and different people react differently also to different types of probiotics.
So not necessarily sufficient scientific evidence, and a lot of variation, and depends on the individual. But the last question talks about, is it possible to consume too much of the probiotics and trigger symptoms of IBD?
I think that probably for everything, there's something that becomes too much. I haven't seen any specific evidence saying that probiotics trigger IBD.
Great, thank you. Next series of questions. What is the
relationship? Oh, sorry.
No, go ahead, please.
There's a really important relationship between the diet and the microbiome. And as we talked about in general, I think that what really for my family, what we're trying to aim for is eating different types of fiber and eating a lot of fiber. You know, one of the big question about including fiber in the diet improve the microbiome. Yes, in general it does. As I mentioned, it's important that the right bacteria are there, because otherwise you'll be feeding something that is not going to be eaten. But in general, it's kind of amazing how our suggested dietary levels of fiber are in the order of 30 grams that are suggested to be healthy. But if we look at what traditional populations do, they take an amount of fiber that is about 5 times as much. You know, they eat up to 150 grams of fiber per day. And again, this is not necessarily what we want to do, because we have a microbiota that is not used to that amount of fiber. But I think that as this research continues, I think we're going to learn a lot more about how we're going to have to improve this diet across our population to improve some of these inflammatory diseases. I'm a very big fan of organic diet. I think it's good for the environment. In terms of actual solid scientific data as how it's good to the health, there hasn't been something that is really strong to show that that it's good. But I'm again, this is a personal opinion. I like organic diets myself. For the question about lactose or gluten sensitivity associated with the microbiome, absolutely. This goes back to the question of malabsorption. So when certain molecules are not being consumed, they're there and they will physically draw water out and they will cause diarrhea. So this is very tightly linked to the microbiome. And for specific diets that have an impact on the microbiome, they all do, because we're just feeding different microbes with different types of diets.
Great, thank you. Next slide. So what is the impact of conventional IBD treatments on the gut microbiome?
There's one of the big impacts is that for surgeries that require antibiotics, there's definitely going to be a change in the microbiome. And you know, this is something that in speaking to patients, IBD patients, I've heard from a lot of them about how after taking antibiotics that things just didn't feel quite right. And of course, antibiotics become really important in some cases and really should be taken. And one of the things that I think that now the microbiome field is really pushing for, and as well as clinicians, is to try to make sure that antibiotics are taken for the right reasons. And in general, also in terms of biologics, they definitely affect the gut microbiome. And one of the things that that I alluded to in the talk is the fact that depending on the gut microbiome, we also know that they might work better or not. So remission is very correlated to specific types of microbiomes being present at the beginning.
Thank you. Related to this series of questions, there was an audience member who asked a question about whether the prep that patients have to undergo before a colonoscopy, whether that has a similar effect in terms of the diversity in population that causes after diarrhea.
Absolutely, that's a great question, and it does. So what we find is that the colonoscopy preps really wash out your microbiome. But one of the things that we found that is interesting and that was unexpected for us was the fact that it actually seems to be more problematic if the diarrhea is persistent. So a one-time colonoscopy prep, it's not enough to really kill off the microbiome. But if you stress it over long enough that it's not able to grow again, that's when things become problematic. And this also happens, incidentally, is the reason why when you take antibiotics, you're supposed to take the full course, because in the full course tends to be over the time span of a week. Because during that time, you really make sure that your pathogen is not able to hide and then regrow. And so from the perspective of our good microbes, sudden perturbations seem to not be catastrophic. They seem to recover from things because there's enough of them to really be able to bounce back. But more chronic, for example, use of laxative, we find that has detrimental effects over the long term.
Thank you. Next question, Sarah. If you can elaborate on this, this is, I think, might be a bit challenging for you, but can the gut microbiome be used to make a diagnosis of IBD?
That's a fascinating question with a not very, I guess, fulfilling answer. The true answer will be sometimes, not always. So what we find is that the microbiome in IBD can be very different, but in very localized locations. So one of what I was telling you about trying to make sure that we know where different species are within the gut becomes really important here, because sometimes the microbiome is going to be very different at places of lesions. And that's really where we detected differences, not necessarily in the stool samples that we would analyze. So in some cases, we can tell that the microbiome is just very different and has a lot of species that are related with inflammation. But sometimes these species hide in the places where they're making the damage. And those are not the large majority that makes it out of the gut. So I guess that's maybe a kind of an unsatisfying answer, but not always can we have it as a diagnosis.
Okay, next question is, how is the gut microbiome linked to mental health in people with IBD?
This is a really fascinating question, and what is called in the field is this gut-brain axis, and we know that there's a very tight link between our microbiota as well as our mental health, and even related to neurodegeneration. This is something that we're really interested in understanding. It becomes really important with the type of products that bacteria make, and this will affect the way that we feel. One of the things that I think that everyone can attest to is that if you're hungry, you're likely in a bad mood. A lot of these different ways in which we interact with our microbiome are through these indirect metabolites and products that make us feel in a certain way. And this idea also that mental health is being affected, I think is one that is really important, and that we're really interested in understanding more, but we're just at the beginning of these studies.
Okay, next question. What is the relationship between strains of bacteria, the microbiome, and IBD? So I know you touched upon this, but if you can elaborate again.
Yeah, so the specific bacterial strains they are very different for different people, in the same way that for different people, eating a slice of pie will make them their sugar levels in the blood go up, and versus other ones that don't. So this is the strains that are going to be good for you are going to be very dependent on your history, on who's already there, and in general, we tend to think that high bacterial diversity is linked with health. But we also know, in some sense, we know more about when bacteria are bad. And we tend to know that there are specific bacteria that have toxins and that reduce gut health and promote disease. But in terms of good bacteria, there's a lot of them, and I think that it really becomes dependent on who else is in your community to know whether you should add those ones back in.
Thank you for that. So I know we talked a lot about bacteria, but it looks like we have a graduate student in the audience who wanted to ask you a little bit about other forms that make up the microbiome. In particular, he is posing a question about phages and whether whether you can comment about phages, whether they're a cause or an effect of IBD, and what kind of evidence there is around phages.
Yeah, so maybe to just give a little bit of an introduction, phages are viruses that attack bacteria. And so they're the most abundant type of entity that is present on our planet. There's an astounding amount. We think that there's about 10 to the 30 of these phages floating around. And one of the things that becomes really important to understand, for example, in the case of IBD, is we know that when the physical environment changes, it also affects not only our bacteria, but also the way that these bacteria can be attacked by these viruses. And one of the things that we're starting to find out is that these viruses change a lot. And now the question has become, what is the cause and what is the effect? So are these phages really the puppeteers that control the bacteria versus are they changing just because the bacteria underlying are being affected? One of the things that I think is really fascinating also is that there's more and more evidence now that a lot of the bacteria that are pathogenic have toxins that come from phages, because these phages go in and they can embed in the genome, in the genetic material of these bacteria, and they can make them produce these toxins that allow them to survive more. So this is really an active area of research, and I think that as with a lot of these biological questions about what's the cause and what's the effect, I think that it's really a bit of both. There's going to be, we know that changes in the environment change the way that some of these phages can infect the bacteria, and therefore are going to change the way that they can produce molecules and affect our health. But we're also going to have the other effect, in which in some cases we're going to have some phages that come in and they will bring in some toxins, which then will trigger the increase of these inflammatory states.
Great, thank you so much. Okay, next question. Are vitamin D levels associated with changes in the gut microbiome and in inflammation of the gut?
And this is vitamin D is definitely a very important vitamin, and it's really associated with the immune system. So what we know is that in the case of low vitamin D, one of the things that happens is that our intestinal barrier becomes much more leaky. And in those cases, increasing the amount of vitamin D can really help reestablish this barrier. So it's very important, and it will definitely affect the microbiota because it'll change the way that we can interact with it.
Thank you. I'm going to switch it up a little bit and ask some questions. There's a number of questions that have been posed by the live audience. So there's one that has to do with your study, related to your study, but applying to the human situation. So one audience member is asking, does the length of time that the microbiome is disrupted have an impact on recolonization? In other words, is it months, years? Are there some evidence that you can talk about?
Yeah, that's a great question, and the answer is absolutely. So the timescales of the perturbation really make a difference in the way that the microbiota can rebound. So one of the things that we talked about is, for example, in the case of colonic lavage, that if it happens over one day, the microbiota, for the most part, as far as we can tell, can recover back. What becomes really hard, and what we're seeing over this generational times, is that if you lose a microbe and then you don't have it around in your environment, it can't come back. You just don't have a reservoir of it. And that's one of the reasons why we're trying really hard to study these populations that haven't been exposed to antibiotics and that haven't changed their lifestyle, to make sure that we have a reservoir of these bacteria as we find out how they might be important for our health. And of course, with each, as I mentioned, which one of these bacteria, we're going to need a lot of research to try to understand whether they're good in general, whether they're good for a subpopulation, and how they can interact with the microbiota that we already have. But I think it's going to be critical, and it's a race against time to make sure that we store some of these bacteria, because if we lose them as a global population, we just don't have anywhere to get them back from.
Thank you. There's another question. I think it's linked to what you said about fiber intake and how the general population takes their intake is quite a bit more than the daily recommended dose. Someone is asking whether it's safe to take Metamucil every day.
I think that's a question that would probably be more appropriate for your doctor. One of the challenges with the individual fiber types is that they will encourage growth of bacteria that can grow on Metamucil. One of the reasons to try to go for more different types of fiber is that even in one type of veggie, there's going to be hundreds of different types of fiber. Another thing that we didn't touch upon, but I think it's important to point out, is we're so tightly associated with our microbiota that in maternal milk, so in the breast milk, about 1/4 of the dry weight is made up of actually types of sugars, which in the end, fiber ends up being just carbohydrates that are stuck together, that the baby cannot consume. So it's just things that our microbes are able to break down. And this is very special for humans. Other types of primates do not have this. Cow milk doesn't have this. And so for humans, it just really is important to keep this microbiota from very early childhood. And the diversity of these different types of fibers and different types of sugars becomes really, really important. And that's why we try to recommend that the diversity is there. And for example, from the perspective of the babies, formula is starting to have different types of sugars that are good for the microbiota. But in maternal milk, there's just hundreds of different types, and we just make them. Again, with the idea of also the natural products, they can really, we've evolved with them. So this is just what has been good for us because this is what we've been exposed to.
Thank you. That's very interesting. I am going to end off with one more question. Again, it might be more of a medical question, but I'm going to pose it to you anyhow. For someone who has a hard time digesting most fibers, are there any particular fibers that you would recommend?
That's a really good question. This is, the best way of finding out is trying with simpler fibers. I might suggest bananas and then building up slowly. One of the things that happens is that as we build up with more fiber, there's a period in which your body adjusts, and sometimes you might have a little bloating, but then it goes back down. So I think if you're starting from little fiber in your diet, you want to take it really slow, and just add a little bit, and then as your body gets used to it and as your microbiota builds up to actually be able to ferment it correctly, then you can slowly build up the amount. And of course, this is, for different people, different types of fiber are going to have different effects. So in the end, each one of us, in some sense, has to try for ourselves to see what works. But I think it's really critical to start small and build up.
Thank you for that. That was fantastic. I think we're near the end. Answered a lot of questions, so thank you so